Good morning, everyone. My name is Alanis-Lobato from the Integrative Systems Biology Lab at King Abdullah University of Science and Technology. My talk is about prediction of genetic interactions using network topology. What is a genetic interaction? Suppose that we are interested in phenotype cell fitness. So we have a wild type with 100% fitness. Single deletion of gene A drops it down to 60%, while single deletion of gene B drops it down to 50%. So the expected phenotype given by the multiplicative effect of these deletions is 0.6 times 0.5 equals 0.3. And a genetic interaction is defined when the double mutation of these two genes generates a phenotype that is worse, called negative interaction, or better, called positive interaction, than the expected. Genetic interaction detection plays an important role in tackling the intricate relationships between genotype and phenotype, understanding human disease, and designing personalized treatment. The problem comes when we want to map every genetic interaction. This task is far to be completed, even in model organisms like worm or yeast. And here's where computational techniques come into play because they reduce costs incurred by the experimental detection of this interaction, point out good candidates that of course reduce the universe of possibilities to test in the lab. Previous attempts to computational prediction of genetic interactions depend on biological information that for some organisms might not be available. So our proposal is the exploitation of the information stored in the topology of the network, which should be there because it was shaped by the biological properties of its components. So how this genetic interaction prediction is done? We have a genetic interaction network, and we want to know if those genes that are not directly connected are likely to interact. So we represent this network topology as an adjacency matrix, where 1 means there is interaction and 0 means there is not. And we input the matrix into a prediction technique that outputs a candidate interaction list sorted by likelihood, which means that at the top of the list we have the best candidates. And to validate our prediction, we do a gene ontology relevance test, which means that if two genes share similar function, are involved in the same process, or their gene products are located in the same cellular compartment, we have gene ontology relevance. And to assign a number to our validation, we iteratively take interactions from the top of the list, and the fraction of those interactions with gene ontology relevance is reported in a precision curve. So imagine that I take the first interaction, it passes the test, so I report 100% precision here. I take two interactions from the top of the list, but only one passes the test, so I report 50% precision. I take three, but only two pass the test, so I report 66% precision, and so on. Usually we are interested in taking from the candidate list 10% of the links in the original network because the ones at the bottom are less likely to interact. And finally, we have that an area under the precision curve is the one that summarizes the performance of my prediction technique. There are two approaches to link prediction in networks. One is neighborhood based, the other one is network embedding. The first one relies on the number of neighbors the nodes that I'm analyzing have. In this case, I have nodes X and Y, and to assign a likelihood score here, I count the number of neighbors they have in common. This technique is called common neighbors. So here I have 1, 2, 3 common neighbors, and I assign a likelihood of 3 to this non-existent interaction. So the higher the score, the more likely X and Y interact. Network embedding relies on the idea that the network is embedded in a high-dimensional space, and I want to map it to a low-dimensional space. And to describe this situation, I will talk about minimum linear embedding, which is a technique that we developed here in our lab. The idea is to extract the minimum spanning tree from the network, compute shortest path over this minimum spanning tree to generate a distance matrix to which we apply dimensional scaling. We map the nodes to the low-dimensional space. We reconstruct the network there, and we compute shortest path over this network to assign the scores.